Hello everyone, it's a pleasure to be here. Today I'll talk about my work on programming with molecules. At the end of this talk, you'll gain an understanding on how we can utilize traditional computer science principles into a biochemical domain, that is how we can program with molecules such as DNA. Also, you'll learn about connections between different models of computation through examples of connections between neural networks and chemical reaction networks. Let's dive in. So why would we even care about programming with chemistry? So one fundamental reason is that we cannot embed electronic logic into biochemical environments, while chemical logic is naturally suitable and compatible with those environments. So why would we want to embed logic into biochemical environments? Well, there are many potential applications that would enable. One, we can design chemical controllers that can help increase the yield within bioreactors. Two, we can help in manufacturing of nanostructures and materials via self-assembly, where we can design tiny molecular structures that, based on local interaction rules that we encode, they interact with each other and construct a larger pre-planned shape. On such a tiny scale, this is the only technique that could build complex shapes. Three, we can help design smart drugs for health, where we can have chemical controllers that can target only cells of certain kind, like infected cancer cells, and they can neutralize only those cells, leaving healthy cells intact. So my approach in programming with chemistry is to look at software engineering, machine learning, and molecular programming from a unified perspective and try to extract common principles and apply between the areas. So before diving into my work on how I design chemical computation, I'll give you an intuition on what it means to compute in chemistry. So let's look at the reaction A plus B goes to C. Indeed, the default language to specify behavior in chemistry are chemical reaction networks, that is, systems of reactions. If we try to interpret this reaction, so we have a molecule of A, let's have a molecule of A as shown here, and molecule of B as shown, and they are compatible with each other, so they can interact and produce molecule C. Now, what does this CRN compute? Well, the reaction can occur as long as both A and B are present. Thus, when one that was initially present in lower quantities, either A or B, runs out, then production of C stops. So the amount of C produced will be equal to the minimum of A and B initially present. And we can say that the CRN A plus B goes to C, thus compute minimum function. Another thing to note is that CRNs can be described, their behavior can be described with a set of differential equations, where the above reaction can be described with this set of equations. Now, you don't have to understand details, but what is important is then this allows you to actually numerically solve this system and see how quantities of different molecules change over time. So as shown here on the plot, we initialize the system with some quantities of A and B, which is 3 and 2 in our case, and this shows how A and B change over time. Because we are talking about programming, we need to somehow define inputs and outputs of our programs. And we take a convention that inputs are initial concentrations of A and B, while what is the output then? Well, output is the final concentration, that is quantity of molecules of type C. So now I just want to stress that encoding desired intent in CRNs is challenging. And let's look at this simple two-reaction system, which is characterized by differential equations on bottom left. Now, if we simulate the system and see how quantities of different molecular types change over time, we can see this complex nonlinear behavior. And what that implies is that if we want to encode some desired functionality into CRNs, it's pretty hard to do that. Another reason for that being hard is that every reaction happens in parallel. So we have a highly and inherently parallel behavior. And we know from traditional programming that as soon as we have concurrency, a lot of trouble arises because we are not used to thinking about things in this highly parallel way. So thus, one goal of my research is to make programming paradigms that abstract away this complexity and makes it easy for a user to describe desired intent, and then from that high-level intent we can translate, we build compilers that translate to the level of chemical reactions or even chemical hardware. So that's a goal. So I would argue that one of the crucial steps in achieving this goal is coming up with the basic primitives or modules that should be used at the root of this language. So basically you can imagine this as instructions, like basic instructions, and then these instructions should allow combining them into some programming paradigm. So I built a technique that enables discovery of chemical primitives. It basically explores the space of chemical reactions and finds primitives. So to do that, I utilized Alloy, which is declarative language for specifying structural and behavioral properties developed at MIT. And in Alloy, I write a specification of what CRNs mean and what class of CRNs we care about. So what Alloy then allows is translating this to a Boolean logic, a Boolean formula, and then providing it to SAT solver. And SAT solver can return all instances, that is all CRNs, all chemical reaction networks that satisfy our model specification. So in that way, this allows us to enumerate all CRNs. For example, CRN1 is this reaction A plus B goes to C, and then SAT solver enumerates all other instances, and we can arrive to some CRN K. And then I should mention that we have another part of our technique, which is analyzer. So what analyzer does, it can take a CRN as input and it can say which function the CRN computes. So what we did then is for all CRNs we enumerated, we ran analyzer and we found some interesting and novel CRNs. So CRNs computing some intriguing functions. So one such CRN that we found is ReLU computing CRN. It computes a ReLU function, which is a default activation function in neural networks, and thus this kind of appealed to us a lot because of the popularity of ReLU. So this work achieved Best Student Paper Award. So I'll now talk about neural networks and chemical computing. So we have a model of neural networks on one end, and on the other end we have chemical reaction networks. So we wondered if there is a connection between the two, and indeed we discovered the connection, and that connection is so tight that a class of neural networks and a class of chemical networks can be considered the same thing. At the root of this discovery is our discovery of ReLU CRN using technique shown on previous slide. So what does this connection bring to us? So basically it builds a bridge. It builds a bridge between neural networks on one side and chemical networks on the other. And now this bridge enables us to utilize deep learning to train a neural network in silico and then utilize this bridge to jump from a neural network to a CRN that has equivalent behavior. So this CRN for same inputs as neural network will produce the same outputs, but now in the chemical world where inputs are concentrations and outputs are also concentrations, that is quantities of certain types of molecules. So this work was published at our ICML and PNAS papers, and now we performed experiments of this technique. So for one such experiment, we had a neural network which on its inputs receives gene expression levels and is trained to predict virus types, so virus that is present, such as H1N1. So we train a neural network which is close to intended application because gene expression levels are naturally available within cells, and the neural network is trained in silico. Then we translate it to chemical logic, and for now showing simulations that the behavior is equivalent. This virus detection. Then we move to spatial pattern formation, another experiment. So for this, actually I should mention that spatial pattern formation has important applications in synthetic biology because tissue and organ engineering rely on it. To model this problem, we have a neural network which on its inputs receives x1 and x2 representing coordinates that is positioned in space. A neural network is trained to predict the pixel or the color, such as black or white. So in this way, you can train a neural network to learn a pattern. So based on a position in space, the neural network will output the color that's present at that position. And now we translate this neural network to chemical logic and show via simulations and proof that behavioral chemical network is equivalent to neural network. But one question remains: can we actually physically implement this? And I'm excited to say that the case is yes, we can, and we did so. So we used this pattern, which represents a UT logo of Longhorn, and we discretized it. Use this discretized version to train a neural network that, based on a coordinate x1 and x2, outputs color value. This is the neural network that we learned, and this is how the output of the neural network looks like. So here, based on the position in space x1 and x2, this is the output of the neural network. And if you discretize this output, if you cut it based on the 0, you will get exactly the same pattern as shown here, as initially trained on. But now, so what's important is that we translate this neural network using the bridge I showed before to CRN, this chemical reaction network, and this is all what I talked about just before. But the new part is that we implement basically DNA molecules that implement this chemical reaction network. So this is how they would look like. So for the CRN, we designed DNA molecules that implement it, and we ran wet lab experiments to see the behavior. These are the results of the wet lab experiments, and they are pretty close to the behavior of the neural network, that is, to the ideal behavior. So in this sense, we successfully implemented a neural network in DNA. So this paper is currently in preparation. Now I just want to stress that DNA has been utilized as a powerful computing substrate where researchers have built plenty of exciting things. So researchers built a cargo sorting DNA robot, then an RNA robot that performs computation inside of cells has been built as well as DNA technique that performs cancer diagnosis within a serum. Now, a couple of words about future work. So I imagine in designing chemical computation that we will have different abstraction layers similar to what's found in programming electronics nowadays. So at the top, I imagine having a high-level programming paradigm which makes it easy for a user to describe desired intent. Then we will have compilers from that level to chemical reaction networks, and this is abstract layer, and as well as compilers from this level to chemical hardware such as DNA. Now there are many open questions at all parts of this diagram. For example, at the top, so what is the right paradigm to program? Should it be imperative language, concurrent, or should it possibly be neural networks as I presented in this work? Then we need efficient tools for basically efficient compilers from these high-level programs to chemical reaction networks. Then we need tools that discover chemical primitives that are good and performant, such as one such technique I talked about in this talk. Then there are open questions in translating from level of chemical reactions to DNA. So basically we need better tools for systematic translation from layer of abstract chemical reaction networks to physical DNA implementation. So we had to do a lot of manual work to design DNA molecules to build that Longhorn pattern I showed, but we need to automate and improve. And at the level of hardware, there is a question of biointerfaces. How do we make this hardware that we build interface with the real-world environment, such as biochemical environment, cells, and so on? And there are different sources of error that we don't fully understand when we design DNA molecules, some spurious reactions, and so on. And also a question of scaling up, basically scaling up the size of the hardware that we can build, or in other words, scaling down the cost of building hardware. So I already tackled some of these questions, of course not completely solved, but here I list them. A CRN++, which is the first imperative molecular programming language as far as we know. I didn't mention in this talk, but you can take a look. Then this bridge from CRNs to neural nets I talked about, as well as CRNs exposed. That's a technique I briefly mentioned about discovery of chemical primitives. So now I just wanted to put this work into a broader context of analog computing. And here I show a bubble sort algorithm that's implemented in analog world, that is with CRNs or ODEs. And what was intriguing to find out that even this simple algorithm that we know very well in digital logic, we had a hard time understanding in analog logic. We had a hard time understanding how it works and why it works. And what this also says is that we know little about programming in analog world. And by building up on our foundational knowledge of analog computation, not only we will benefit for computing with chemistry and CRNs that are inherently analog, but some other areas might benefit as well, because we know that analog computers are much more power efficient, and there is recently interest in building analog computers that execute neural networks to draw down the costs. So contributions in molecular programming that I made is that I designed a technique that systematically explores space of CRNs, using which I discovered extremely compact ReLU computing CRN and achieved Best UN Paper Award at DNA conference. I designed first imperative molecular programming language, CRN++. I discovered a tight connection between neural networks and chemical reaction networks using great independent scheme. I was involved in DNA wet lab experiments project, implementing and experimentally verifying our neural network to CRN scheme. And I argued importance of examining other forms of computation, such as analog, and building a holistic view unifying distinct kinds of computation. So as far as we know, there is 3 kinds of computation happening on Earth. 1, computation within our computers. 2, computation within neural networks of our brains. And 3, computation happening within every cell of every living organism on Earth within complex gene regulatory networks. In my research, I explored connection between these models, and I believe by building a holistic view on computation, we will get fruitful results. Finally, I'm glad you're listening to this talk, because by listening, you start contributing and raising awareness of importance of emerging unconventional computing methods that will likely yield very exciting and novel applications.